we're going to go through the endocrine histology that you'll cover in lab. To begin with, you'll look at a cross-section of the pituitary glands. Now, of course, there's an anterior and a posterior pituitary, but we prefer for you to identify them as the adenohypophysis, seen here, which is the anterior pituitary gland, and the neurohypophysis, seen here, which is posterior pituitary. Neurohypophysis gets its name from the fact that the cells here, which stay in a lighter pink, right, don't look quite as clustered as what's seen in the adenohypophysis. These cells are very similar to the astrocytes of the nervous system. Right? So they're not actually going to be producing hormones, rather they receive and store those that are made by the hypothalamus. The adenohypophysis, however, the cells here, larger, more like glandular cells, like what you would expect to see in the rest of the endocrine organs, do produce those hormones that they are known for. So that's where you have your FSH, your LH, your ACTH, your TSH, as well as prolactin and then growth hormone. So please note each of the hormones made by the adenohypophysis, and note that the neurohypophysis is meant to receive and store both your oxytocin and your antidiuretic hormone. As we move to the thyroid slide, you'll notice that this actually shows the thyroid and the parathyroid glands together. They're quite distinguishable when you take a look at it here at 40x magnification. Note that the cells of the parathyroid gland are pretty densely packed together, right? And they're much smaller. When you look at the thyroid gland, you can tell the difference here by the presence of these follicles. Let's focus in here on 400x. This is looking specifically at the thyroid gland and one of the follicles that you see. The type of cells that actually line the wall of the follicle are called follicular cells. And these follicular cells are going to work in conjunction with the colloid, this material in the center, to produce your types of thyroid hormone, right? T3 and T4. The cells that neighbor the follicles and run right in between them, right, that do not touch colloid, that's an easy way to think of it. These cells, called parafollicular cells, run between follicles and do not actually make contact with any colloid. Those are making calcitonin. If you go back here and you look at your 40x representation, knowing that calcitonin and thyroid hormones come from the thyroid gland, you can then remember that parathyroid hormone is what's coming from the parathyroid glands. The adrenal gland is next. The adrenal gland is a little bit more complex than the other organs because of the different layers that you have to identify in a cross-section. We'll start here on the most superficial layer. This is the capsule. It's just going to be a connective tissue layer that wraps around the organ for protection. We are not talking about any hormones that would be secreted there because that's not its purpose. The first layer of what's known as the adrenal cortex, seen from here to here, this is all adrenal cortex. The outermost layer is the zona glomerulosa. The next layer below that is the zona fasciculata. And then below that is the zona reticularis. So let's examine the differences between these. The zona glomerulosa, if we move to the next slide, when magnified, you can see the cells are going to be wrapped in little bundles or clusters that are slightly rounded. So if you think that the letter G looks rounded in nature, then that can help you remember that glomerulosa has the rounded clusters of cells. This is where you're going to see mineral corticoids like aldosterone being released. Zona fasciculata is going to be made up of cells that are slightly larger, so you can see how they have quite a bit more cytoplasm and it stains a little lighter. And these cells are going to be lined up in cords. You can actually see that a little bit better if you look here. Notice how each one of these fascicles or long cords runs straight up and down. And again, you can see that here. Fascicles are cords where the cells run straight up and down. So this is different from that round cluster appearance that you see in the zona glomerulosa. This is where you'll start to see glucocorticoids, right, as well as your gonatocorticoids being released. Now the reticularis, we have a nice image right here. Because of the expanse that you see, right, you can note the fact that it doesn't have that characteristic fascicle or parallel cord look that you see in the zona fasciculata. Instead, reticular means net-like, and so you can kind of imagine that there's no true pattern to any of these cells. It's just like a bunch of random fish caught in a net spread out everywhere, and so that's what you see right over here. They refer to it as a branching kind of an orientation. Once you've identified each one of these as the adrenal cortex, then it's easy to distinguish them from the innermost region called the adrenal medulla. The adrenal medulla is going to be secreting neurohormones like norepinephrine and epinephrine, but the cells that are here are more representative of the nervous system than they are of a gland, like you would see through the rest of the adrenal cortex. The cells that are here are a lot like neurons that just don't have dendrites or axons. We have often referred to the adrenal medulla as a postganglionic sympathetic neuron cluster, and that's really what it is. You're going to have a preganglionic sympathetic neuron that targets that region, and it is going to be the cause of hormone or neurohormone release into the bloodstream. The pancreas is the next organ, so this is going to have a combination of exocrine and endocrine function. The majority of the cells that you see are referred to as pancreatic acini, right? Acini is plural. A single cell would be referred to as an acinar cell. All right, ACINAR. And these are going to be producing the digestive enzymes and other secretions that are associated with digestive function. And each one of these will be secreted into a duct system and deposited within the small intestine. You'll also see light staining patches here and there referred to as pancreatic islets, right? So they're islands in the midst of this large pancreas. And they really are the minority, so you only really see a few of them whenever you're looking at 100x magnification at the pancreas. So we're going to zoom in right there so you can see the difference. So here's a nice pancreatic islet with all the cells that are within, and then here's some nice view of all these pancreatic acini that surround it, so you can see pretty clear difference between the two patches. The pancreatic islets are where you contain your alpha, beta, and delta cells. Alpha cells are what produce glucagon, beta cells are what produce insulin, but as you can see here, it's very difficult to distinguish. We actually don't have the appropriate stain type to make that really characteristic. So we will not be asking you to identify alpha or beta cells. Just understand that you have both glucagon and insulin coming from the pancreatic islets. The next organ we move to is the thymus. The thymus itself is going to be made up of two large lobes that is then divided into individual lobules. So lobules are very small lobes within the thymus. And you can see how they're divided right here by these nice divisions referred to as trabeculae. So trabeculae are these branching divisions that separate individual lobules. As we start to move through more organs this semester, you'll continue to see this designation of cortex and medulla, just like we saw with the adrenal gland. And typically the cortex refers to the outer layer and medulla refers to inner regions. But in an organ like the thymus, you can see that the medulla is actually going to be scattered throughout the cortex. So anywhere you see all these dark clusters, that tends to be referred to as the cortex, sometimes called cortical regions. Medulla are going to be these light staining regions right through here. And the cortex is just going to look darker because it has a more dense cluster of cell types. As we zoom in right over here, you can actually see once again the difference between medulla, right, or medullary regions, and the dark staining cortex. And again, you can see here's one of those nice trabeculae dividing lobules, and within it I want to point out one of these arterioles. Now, you're going to see lots of clusters of different cell types like we saw with the pancreas and the pancreatic islets. As long as it is solid, right, then it's going to be some kind of a cell cluster. But if it is nice and round or oval shaped and it's hollow, that's a blood vessel. You haven't learned how to distinguish arterioles from venules yet, but note that this is an arteriole that you see. If we zoom all the way into 400x, we're actually looking at a region of the medulla right here. You can see a number of different cell types. The majority of what we see here in this image are lymphocytes. Lymphocytes have varying functions and are tied to the immune system. And then you're also going to see these larger, darker staining reticular epithelial cells. Now this is where our endocrine function tends to come from. These are the cells that are going to make your thymosin, your thymopoietin, and your thymulin. We're also going to see that they secrete different chemicals to help with immune function like interferon. But for right now, recognize those as the production site for your endocrine function. Now, of course, those secretions, like the thymulin and the thymopoietin, those are going to be responsible for helping with the development of the organ along with the development of all these lymphocytes. As long as you have the appropriate levels of those hormones coming from reticular epithelial cells, you can develop a better immune system that's more likely to target what is needed to. You'll also see a variety or a handful here of macrophages. Macrophages tend to have this nice oval shape. You can see another one right through there. Their cytoplasm stains more lightly than the lymphocytes will, and they tend to have this teeny tiny little dark nucleus. Macrophages, they are phagocytic cells, and they can eliminate some of the pathogens or the debris of dying cells from this tissue. Now, one of the structures that can be very difficult to find, so you may want to ask for assistance with, is called a thymic or a Hassel's corpuscle. These are going to be collections of reticular epithelial cells. So you can see this nice pink appearance on the outside and then get an idea for how that pink is going to factor into one of these bundles. This one looks kind of elongated. Sometimes they look nice and round. So there's a high level of variety here in how to identify these corpuscles. Their identity is not, or their function is not well understood. But being able to recognize them can help you classify this as the thymus. You can actually see some of them when you zoom out to 100x magnification. You want to look for things like this, right? These large clusters. Those are definitely going to be reticular epithelial cells. And as long as there are multiple reticular epithelial cells clustered together to look something like this, then that's what you would technically refer to as a thymic corpuscle. With the reproductive organs themselves, the testes and the ovaries, these are important because not only do they respond to hormones that are secreted by the rest of the body, but they're going to make their own hormones. So to begin with right here, a cross-section of the testis reveals the appearance of these large hollow tubes referred to as seminiferous tubules. These tubes themselves are where you see sperm produced. So the sperm originate as these tiny cells on the border, and then as they develop tails, they actually move here to the center. Here's some nice tails of spermatozoa that are developing here. When you have follicle-stimulating hormone, FSH, secreted by the anterior pituitary, this is the region of the organ that FSH targets. And it's actually going to cause those cells to proliferate and then to begin their nice long journey. Right. In between the seminiferous tubules, you see clusters of interstitial cells. So inter meaning between. Right? Interstitial cells are producing testosterone. And testosterone is very significant for secondary sex characteristics of males, as well as promoting, in addition to FSH, promoting the development here of the spermatozoa within seminiferous tubules. The testosterone will be released whenever luteinizing hormone from the anterior pituitary travels through the bloodstream and targets these cell clusters. For the female reproductive system, we look at a cross-section of the ovary, and this is a pretty close-up look. This is 100x magnification, and there are two things that you see here. Once again, you see follicles. So again, if a follicle is a package, much like we saw in the thyroid gland, then each follicle is going to be multicellular. Recall that in the thyroid gland, it was the same way. Lots of cells, right, forming the border around something in the middle. In the ovary, that something in the middle is an oocyte, right? This is it. It's giant. So this is one cell right here with all of its cytoplasm and its nucleus, right? Here's another one right there, giant cell compared to what else you see, oocytes. These are developing ova. These are what will eventually be ovulated and then are available for fertilization. Now the follicles themselves are going to range in size, so some will be teeny tiny and some are going to be giant, right? But here's what they have in common. The follicle cells are what respond to FSH from the anterior pituitary, and in doing such, they'll start to proliferate, and so a follicle which is small right over here can eventually become large like the one you see here. The second thing you need to know is that these follicle cells are actually going to be producing estrogen, which is what's responsible for secondary sex characteristics of females. So of course, as the follicles get larger during a monthly cycle, right, the estrogen levels begin to rise. The oocytes themselves stay contained within the follicle and eventually will undergo ovulation when you have a surge of luteinizing hormone from the anterior pituitary. And that's going to require some additional processes that we won't study until later, but you should understand that the ability of the oocyte to ovulate, right, and to actually leave the ovary, leave its follicular package, will arise whenever luteinizing hormone is secreted. So that's your endocrine system and the histology there. Make sure that you identify cell types if named, and make sure you identify tissues if named, as well as the name of the overall organ. And the last thing you want to be able to do is tie in all hormones that are associated with each tissue we examine.